Hi everyone, and welcome back to our Building Genomics Informed Conservation session. So kia ora koutou from me from New Zealand today, and as we just stated already, I'm going to chair the second part of this session. And as beforehand, please feel free to ask questions during the talk. So as soon as a question comes to your mind, just pop it into the chat, and please also indicate the name of the speaker that you would like to ask the question to. And then vote up the questions as you have probably done previously a lot by now. I will then ask these questions at the very end. Also, to already mention now, we will have a spatial room for networking right after the session, where all of our speakers from the first session right now and from the second session now are going to be available to answer more of your questions and to discuss in more detail. But now let me introduce the 5 speakers of this session. So we will firstly have Caroline Hogg from Sydney, who is going to talk about bilbies, their importance for Indigenous culture, and the extensive amount of genomic data her team has produced and analyzed so far. We will then have Greg Brimmer from Helsinki, who will give a talk with the intriguing title, Doing Conservation Genomics So You Don't Have to Use Genomics. So he's going to talk about the evolution of Atlantic salmon and how he developed diagnostic markers for conservation of wild populations. We will then have Enrique Figuero, who will talk about the highly endangered black-footed ferret, and his research at the Smithsonian. So Enrique compared various population genomic parameters across wild, captive, and reintroduced populations. Next, Ellie Armstrong from Stanford will report on the first phase of the African lion genomes project to promote the conservation of this very iconic species. And lastly, we will even have a talk about a critically endangered plant species. So Olivia Mendibel Ramos from Cold Spring Harbor is going to talk about the Wollemi pine genome assembly and how they are using long-read nanopore sequencing and also integrating this with expression and methylation data. So as said, please enjoy the talks, ask your questions, and stay on for afterwards for the actual answering of the questions. So off we go.
Good evening, everyone, and thanks very much for having me today. So I'm Caroline Hogg from the University of Sydney, and I want to talk to you about the Bilby Genome Project. So before I start today, I'd like to take the time to acknowledge the First Nations and Indigenous peoples on lands we meet across the world today and pay respects to their ancestors past, present and emerging. So what is a bilby? Small animal, about 1.2 kilos, with very large ears and really known as the Australian Easter Bunny. But more importantly, it's culturally important. It's culturally important to a wide range of Aboriginal nations across Australia, as seen by the variety of different names that are used to describe bilbies. And bilbies are very big and important to the different storylines. And the image you can see on the screen here is in fact the sacred site for bilbies of the Kirakira community, which is one of the Indigenous communities that we're working on this project. Not only are they culturally important to Aboriginal Australians, but they're also biologically unique. So bilbies are in fact the only remaining member of the marsupial family. They're also known as ecosystem engineers. So what does that mean? Well, you can see a burrow here on the screen. They dig large amounts of burrows through the sandy desert regions in which they live, aerating the soil, permitting other species and plants to be able to grow in those areas. As a marsupial, they do in fact have a pouch, but it's a backward-facing pouch, which is very similar to all the other digging marsupials that we find across Australia. They're also omnivorous. They eat a wide range of seeds and insects across the desert regions. And more importantly, they have what we call a karyoaleontoic placenta. And this is only seen in both bilbies and bandicoots. And it's a link, we believe, between marsupials and pouches, more towards the placenta of what we see with the uterus of the eutherian mammals. They also have a very low standard metabolic rate. And most marsupials, in fact, have a lower metabolic rate than eutherian animals. And bilbies have the lowest metabolic rate that's been documented in any of the marsupials to date. They also, although they live in a dry, arid climate, don't consume water. So what is the conservation status of the species? Well, the Yulara, or the lesser bilby, in fact went extinct in the 1960s. And so now there is only one remaining species left in the entire marsupial family. And this, of course, is the greater bilby. And we call the greater bilby the Ninu because that's the term that is used by the Indigenous community that we work with. And the Ninu existed across Australia, both in tropical, temperate and arid regions historically, and now their range has contracted to, by more than 80% across its range due to invasive pests such as foxes and cats. And although they're listed as vulnerable, we don't actually know what the current status of the wild populations are. So this is where the Bilby Genome Project came in. So we did the reference genome from a female using a variety of different sequencing technologies. We have 12 transcriptomes from that reference female, as well as a testes transcriptome from a male. We then went and also resequenced 12 Ninu samples from 6 from a temperate region down here in South Australia at the bottom in the purple of the screen, and 6 animals from the arid region up in Queensland. We also went ahead and resequenced 4 Yulara or extinct species, and these were actually caught or trapped in between 1890s and 1930s when those samples were collected. In addition to that, we've got about 335 individuals with reduced representation sequencing data from the 13 locations of the safe havens across Australia. And these are animals that are on islands or behind fences and in zoos. And also 10 individuals from the Pilbara region, which is the common region up in the northern part of Western Australia where we find bilbies in the wild. And more importantly, we're working with the Indigenous communities to collect scat samples from the different wild populations to help inform them how they're going to manage these species into the future. So what does the bilby genome look like to date? Well, at the moment, we've got a fully assembled genome with it's about 3.6 gigabases, which is quite large for a mammalian genome. And chromosome 1 is in fact 986,000 megabases long. So it's a very large scaffold. And if you look at the transcriptome data, we have quite contiguous genome, and we have about 28,500 protein coding genes for this species. So currently at the moment, we've got the assembly and the annotation started, and we're starting to do a more comprehensive gene analysis. So we're doing an analysis, manually annotating the immune genes for this species, as our research group has done with many other marsupials to date. We're also very interested in assessing the reproduction genes, particularly in relation to understanding the evolution of the karyoalliantoic placenta and how that is potentially a link between marsupials and eutherian mammals. In addition, we're also looking at a number of different taste receptor genes to get a handle on how these bilbies are actually feeding and searching for their food in the arid environment, in addition to some of the other analysis, as well as a cafe analysis, to try and get a better handle on where we're potentially seeing gene expansions for this species. So to date, we've also been undertaking some demographic analysis, and as you can see, there's a PSMC analysis, which has been done by Fred Jaya in Simon Ho's lab, and Carl Erhardt, working with Nathan Lowe, has been working on the runs of homozygosity. And when we look at a comparison between the bilbies in the arid regions and the temperate regions, we are seeing differences. We have seen the fact that there is a difference in the effective population size of bilbies across the surface temperature of the Earth. And we saw a very large significant decline that you can see on the screen with a dark grey area on the PSMC analysis, which is actually indicating the last glacial maximum. For the runs of homozygosity, we do see runs of homozygosity in this species, but they're not very long. And we think this is likely an artifact of the fact that bilbies are a boom-bust species. So what we find with this species is that populations will grow significantly into large population sizes during times of high resources, when it's been very wet and lots of food around. And in periods of drought, the species actually crashes quite significantly and retracts rapidly. In addition to that, we also, bilbies breed every 6 months, and they produce between 1 to 3 offspring every 6 months during the high resource periods. And for that reason, they can have a very high turnover rate in their generation time. In addition to those demographic analysis, we've also been doing a population genetics analysis, assisting with the metapopulation management for the species across the entire range. And as you can see in the PCOA plot here, there is quite significant population differentiation between the different sites across the Australasian region, the region of Australia. And that's, we used about 11,500 SNPs across these 13 different populations to get a handle on what's happening with the current metapopulation. We're very excited to see that there's quite a large amount of genetic diversity in the species and population differentiation, as most of the metapopulation actually came from the original captive population, which commenced back in the 1980s. We are, however, seeing some inbreeding starting to accumulate at some locations. And as part of this project, we've been working very closely with the conservation agencies to make sure that they commence translocations to maximise diversity for the species over time. More importantly, the conservation application for this species, we've developed a number of sex markers already, which allow us to detect the sex of the scat sample that's been collected. And we're now developing a mass array technique, which will use between 50 to 150 SNPs and allow communities to actually start to use the scat samples to be able to investigate their key questions. So they want to know what there's gene flow between the different populations that they manage, whether their management actions have been effective or not in relation to fire regimes and invasive pests, and also for some of their long-term monitoring. And as you can see on the screen here in the top photograph, there's an image of a community rangers collecting the scat samples and putting out a camera trap. But more importantly, the community rangers have been taking out the high school kids, teaching them the Indigenous songlines and storylines, which are really important for them culturally, but also helping getting them to collect the scat samples, which we use for the genetic analysis. So definitely a perfect marrying together of both Indigenous and Western science. So I am not alone in this production of this massive genome project. Here's just a few people who are on the call this week for our genome project, and I'd like to acknowledge all the universities who participated, as well as the Kirakira community rangers and all the community who've been very supportive of the work, and the Bioplatforms Australia and the Australian Research Council for their funding support. So thank you very much.
Hi everybody. As a part of the session on genomics-informed conservation, I'm going to be focusing on a conservation challenge where genomics plays a key role. And this is when in the identification of adaptive loci, because having dense genome coverage is critical for identifying genome regions where the adaptive trait is controlled by just one or a small number of loci. Without high coverage, we may not necessarily have loci in the area where the trait is in the genome. But the point I really want to emphasize today is that once you manage to identify the adaptive locus or a small number of loci, you then don't necessarily need to continue using genome-wide approaches, but instead, for monitoring and management, you can develop so-called diagnostic markers, which focus on those adaptive loci only and are much less expensive than using whole genome approaches. So we've been using this approach in Atlantic salmon, where the adaptive trait of interest is sea age at maturity, or the number of years that each individual spends in the marine environment feeding before it returns to the river to spawn. Now, as most growth occurs in the marine environment, age at maturity correlates strongly with its size, and individuals double in size for every additional year that they spend at sea before they return to spawn. Here's a couple of examples, or real-life examples of individuals that have spent either 1 year or 3 years at sea, and you can see the big size difference there. This age at maturity trait has applied relevance also. These large, late maturing individuals have been decreasing in frequency in recent decades, but they're actually very important for fisheries and tourism because people want to catch fish, and they also have very important role in Indigenous Sami culture and identity. So we were asked by managers to investigate whether the phenotypic decrease in the age and size of maturity had a genetic basis, and if so, what are the environmental drivers of that change? So to do that, we conducted a genome-wide association study for this sea age at maturity trait. We used a SNP panel that had 220,000 SNPs in it, and we're able to identify one single genome region here on chromosome 25 that was very strongly associated with the sea age at maturity trait. And in fact, one SNP in this region explained 40% of the variation in age at maturity, with a more than 1 year difference in the average age at maturity between the alternative homozygotes of the SNP that's near this VGLL3 gene. Here I'll be referring to the E allele for early maturation and the L allele for late maturation later in the talk. So now that we knew now that we had identified the adaptive locus, we were able to develop, we went to the next phase of developing a diagnostic marker that we could use for conservation and management. And then we used this diagnostic marker to look at temporal trends and the drivers of genetic and phenotypic changes in this sea age of maturity trait in a salmon population from far north Finland, which is the source of the data of this graph that I showed you before, where this large late maturing phenotype had been decreasing. So we had this diagnostic marker panel where we included a total of 170 loci. All of the red dots here included several markers for the adaptive locus of interest, as well as a spread of loci across the genome that we could use, that we could amplify about 4,000 individuals for about €5 an individual. And we used this panel to screen about 1,300 samples from a 40-year time series that had all been collected from the same location in the river, but every year for 40 years between 75 and 2014. We also got the phenotypes of the individuals for their sea age of maturity from the scales, which was also the source of the DNA. And what we're able to show is that, in fact, that the late maturing allele frequency had decreased by 18% over the 13-year period. Each dot here represents the L allele frequency for each year. And this change, indicated by the red dot here in the small graph, was the largest change of any of the 170 loci that we screened, strongly suggesting that this can't be explained by genetic drift, but it's much more likely that it's evidence of adaptation. Then we were interested to try and see if we could identify any environmental drivers of this change. So we got together a whole bunch of ecological and environmental data, including prey species abundance in the Barents Sea, the sea temperature, and various measures of fishing effort. And we were able to identify two significant effects. The first was that there was a positive effect on the VGLL3 allele that correlated with capelin abundance, one of the prey species, and also that there was a positive effect on the late maturing allele with the number of nets that were used in the local and indigenous fishery. Just to focus in on one of those changes, the link with capelin abundance, it actually seemed that there was an indirect effect of capelin harvest. So capelin is fished commercially, and when we included capelin harvesting in an ecosystem model, we were able to show that a model that included capelin harvest explained the VGLL3 allele frequencies better than when it was excluded. So this suggests that capelin fishing is indirectly affecting the VGLL3 allele frequencies of a wild Atlantic salmon population. The interesting sort of final point is that the reason that why are capelin harvested, and that's it's a protein source for domestic animal fish food. One of those domestic animals is aquaculture salmon. So we've been able to identify an indirect means by which aquaculture salmon seem to be affecting this wild salmon population. So just to summarize, I hope I've been able to convince you that it is worth the effort to go on from an interesting GROS result, develop diagnostic assays that can be used in conservation and monitoring, because by doing this, we were able to identify several different types of evolution, both direct and indirect, that seem to be affecting this population, which suggests that salmon are in fact adapting to changes in prey dynamics and fishing. But it's also suggesting that the late maturing phenotype is being selected against at the current time. So I'd like to thank you all for listening. We'd like to thank the numerous fishers that have provided scales across this whole 40-year period of the study. These are the first authors of all of the from the papers of the data that I presented. You can find more information about these here. And then I'd like to thank the collaborators and funders. And thanks to you for listening.
So, hi everyone, my name is Henrique Figueiró. I'm a postdoc researcher at Smithsonian Mason School of Conservation. And today I'll present our preliminary results about the Black-footed ferrets genomics project. focusing on population and functional genomics of these highly endangered species. The black-footed ferret was almost extinct by the late 70s and early 80s due to habitat loss and the symbiotic plague that was a disease that was really bad for the species back then. So our objective is to evaluate changes in genome-wide diversity in breeding and drift and breeding load, and focusing on the various variants of both captive and reintroduced populations of the species. We have sequenced 54 genomes, 6 from wild individuals from the late 70s and early 80s, 26 captive individuals, 22 reintroduced individuals. And after mapping, filtering and mapping, we did several diversity metrics, population structure analysis, and runs of homozygosity and both SNP density and the diffuse variants identification. Population structure analysis shows that wild individuals had much more diversity and occupy a bigger space than the ones captive and reintroduced that we have now. For the genomic diversity results, we observed a really low diversity, one of the smallest diversities in mammals overall. And from the plots you are seeing, we see that the mean heterozygosity for wild individuals is much higher than for the captive and reintroduced ones. Here is a plot that I really like. It shows the mean heterozygosity through time. So based on the collection date of those individuals, we see that wild individuals in the late 70s and early 80s had much higher heterozygosity than the ones from captivity and reintroduced ones. This plot shows the mean heterozygosity and the runs of homozygosity. And one thing interesting to show here is that the mean heterozygosity is really low, but the runs of homozygosity reflects a lot more what's going on in the genome, with blocks of bigger than 25 megabases in most cases actually, and the wild individuals having much smaller blocks than the other ones. Here just show you 2 examples of runs of homozygosity in one wild individual and one reintroduced individual, and we can see that the wild individuals have a lot less runs of homozygosity overall in the genome than the captive and reintroduced ones. That's not to say that was nice back then, but it's much better than now. Here is a SNP density plot. It shows the density of SNPs throughout the genome. Warmer colors indicates more SNPs, and then we looked for what genes are located in those regions with higher density SNPs. And what we observed was that we saw a lot of olfactory receptor genes, which expected for mammals, especially for carnivores that hunt based on smell. So that's interesting to observe in the genome we have. Another interesting result we are just starting to analyze is the diffuse variants we observed throughout the genome. So for this analysis, we filtered loss of function mutations that were present in all samples we had, or better, 95% of the samples we had. And what we observed was a lot of genes associated with reproduction, development, and immunity. And so in the future, we'll be analyzing this with more depth, comparing the results and adding more samples. So our take-home message for this study is that genetic diversity is important to assess population health, but more important than genetic diversity is functional diversity that can help you to understand potential health issues in conservation challenges, mainly on highly threatened species like the case of the black-footed ferrets. With the improvement of genome sequencing tools, we can use these tools that were developed for model species in non-model organisms, and that's great for studying biodiversity overall. I'd like to thank everyone involved in this project. There is a lot of people both in the field and in the lab also all the institutions involved in this project. Thank you.
Hi, everyone. Thanks so much for being here today. My name is Ellie Armstrong. I am here representing Stanford's Program for Conservation Genomics. And today I'm going to introduce you to the African Lion Genomes Project, which is work I began during my PhD. We have a limited amount of time, so I will just jump right in. Like many other organisms, lions have undergone catastrophic declines during the Anthropocene. In total, they have lost over 40% of their individuals in the last 20 years alone and occupy only 5% of their historic range. On the left here, I'm showing a map of this range contraction with the historic range in orange, recently extirpated range in yellow, and the extant range is shown in green. While some select populations are listed as critically endangered, the African lion is generally listed as vulnerable by the IUCN. However, they have also disappeared entirely from 12 sub-Saharan countries in the last few decades alone. These losses and subsequent differential management techniques across their range has resulted in a few stronghold populations, most of which are located in Tanzania, but with many isolated and small populations fragmented across the continent. Because of many of these declines, lots of countries and organizations have started to implement translocation strategies or assisted migration into their management plans, such as in the managed metapopulation of lions in South Africa. However, many of these translocations have been made with little regard to the genetic background of the individuals being moved and with little knowledge of the potential for consequences like outbreeding depression or genetic homogenization across their range. Here, I'm just showing one example of a recent and successful relocation effort in Gorongosa National Park that was highlighted by National Geographic, where lions were moved up from a mixed stock of lions in South Africa to Mozambique to repopulate that park after a period of conflict. There's been almost no genome level studies on lions to date. I think there's been about 10 or so low coverage genomes published in total from across their range out of approximately 100 extant populations. And most of this sequencing has focused on the southern and eastern parts of their range. So why focus on lions when I've just told you about how they are a geographically very widespread and complex species that cross many political and country borders? I probably don't have to convince you, but maybe this picture will, that lions are very charismatic. We know this from the Cecil the lion debacle that was recently in the news, but also because they are one of the most used species for logos and marketing campaigns. And this generally is a good thing for garnering the interest and support of the community and to demonstrate the power of conservation genomics. So we can sort of use lions as a flagship species for novel conservation tools in this case. But in addition to being charismatic, like many other large carnivores, lions are a really important indicator species of a functioning ecosystem. So protecting lion habitat is a good strategy for protecting all of the animals and the flora and fauna of the biomes they reside in. And last, it's a species that can immediately benefit from genomic tools that will aid in issues like human-wildlife conflict, poaching, and the illegal trade, where everything from identifying problem animals to assigning a confiscated sample to a population of origin using DNA technologies would help us focus our conservation efforts immensely. So we started out on an ambitious project to map what we call the Genescape of lions, a cartoon of which I'm showing here on the right. The objectives of the project were to first use this data to build a comprehensive idea of the structure of historic and modern lion populations, to then use that information to assess and help maintain this diversity in the future, so to inform things like the translocation events I alluded to before, and also provide a baseline for future genetic surveys. And last, and probably most importantly, to create a database where this information can be easily accessioned by on-the-ground practitioners, whether that is for estimating the number of individuals in a population or for figuring out the origin of an individual during a poaching event. These tools are very important because they then take this project from being a strictly academic endeavor to having direct implications and possible uses on the ground. So you can think of this as like a 23andMe, but for lions. So to do this, we had to gather a large data set of lions from across their range, and this was not something that we could or should be doing alone. So we went to the lion research community for help and founded a consortia specifically for the project. What's great about the lion community, and is possibly reflective of the species' social behavior in general, is that there are already groups formed, like the African Lion Working Group, that bring together relevant governmental, research, and nonprofit entities from across the Lions Range to work on and discuss any issues relevant to lion conservation. So I'm just showing some of our collaborators here, but I do want to emphasize how important this process has been because it has given us a lot of insight based on things like personal observations about potential local gene flow in lions, disease outbreaks that have caused population collapse that haven't necessarily been broadly reported on, and also a path from any results we get directly into local management decisions. So by working with the Tanzanian government, the Kenya Wildlife Service, as well as the Namibian government, we can really kind of get in from the outset and connect those objectives directly into policy. And we're grateful to them for guiding the group's objectives. To start our project, we also built the first chromosome level assembly for the lion a few years ago now, which I'm sure as everyone here has experienced, or if you haven't, you might soon. As soon as we released it, it was kind of outdated since the technology is advancing so quickly. I've just listed some of the stats for those of you who are interested in that kind of thing on the side here. And it is pretty good. It's in chromosomes, but it definitely could be better. So this is something we'll be looking to improve in the future. So now to the more exciting stuff, the samples, what our collaborators were able to put together. We were able to collect over 1,000 lion samples from over 80% of the lion range countries and representing about 60% of the unique extant populations. So far, we've sequenced 500 of these and we have about 200 more on the way. I'm leading the analysis of the continent-wide diversity patterns and our team is doing the R&D for the database and tools, but we do have several data sets which will be analyzed by other students and researchers from their lion population of interest. Like in Namibia, we have a group that's interested in the desert adapted lions, as well in South Africa regarding the captive conflicts going on there. So we have come a long way, but we hope that we can fill in we hope that we can fill in these gaps and kind of expand our collaborations. And we do want to emphasize that in an ideal world, the sequencing would be happening in the same place that the samples are collected, but this isn't possible in many of these countries. But our goal is to make this data completely accessible, not just through these huge FASTQ files that you can download on something like NCBI, but truly accessible to the conservation space through the database and the tools that we're building to accompany the data set. And I just want to very briefly mention that we are using the same framework that we built for the lion, and we've expanded this into a project on snow leopards as well as African wild dogs. We have stellar collaborators for both of those projects as well. And we did pilot a lot of these kind of initial ideas for the SNP panels in the tiger. We have some publications out on those and we'll have more coming out later this year on that effort. And with that, I would like to say thank you to our funders, to you for listening, and to the organizers for such a stellar conference lineup. I'm very excited to hear everyone's talk. I'd like to thank our team leader, Dmitry Petrov, my colleagues, Simon Morgan and Katie Solari, and our broader set of collaborators. We look forward to your questions and discussions during the Q&A, but you are also welcome to reach out to me on Twitter or via email. Thank you.
Hi everyone, my name is Olivia Mendivil Ramos. I'm from Cold Spring Harbor Laboratory, and today I'm going to be speaking about genomics of the recently endangered Willemia pine, Willemia nobilis. So a little bit about background. So Willemia nobilis, it is a living fossil, and that means that it's a taxa that allegedly display extraordinary levels of morphological stasis over geological times. As you can see in here, the current foliage and current reproductive cones, they look remarkably similar to this fossil records that they dated from the Cretaceous. Not only that, Willemia nobilis is a key species to understand the molecular mechanisms of climate change survival. And as you can see in panel B, if you drop all the way from Willemia all the way to the Cretaceous, there has been remarkable changes in temperature and CO2, making this species really key to understand this climate change. But I'm here to talk about why this is an incredible example for conservation genomics. So Willemia nobilis has been declared by the IUCN as a critically endangered species in 2011. But that in combination with the dropping cost of sequencing in the later years, especially once the long-read sequencing platforms came into play, like PacBio and Oxford Nanopore, has dramatically dropped the cost in that sequencing, allowing for very complex genomes like plants and gymnosperms to get fully sequenced and trying to democratize this. Onto some of the results that I wanted to show you today is that the sequencing of Wollemia nobilis has not been an easy ride. It has been remarkably difficult at times, especially during times of COVID, but nothing of this would have been possible without our collaborators in the New York Botanical Gardens. And specifically, they have provided us with a number of tissues to make this project available. In particular, today, I'm going to be talking about the whole genome sequencing that we perform over in the leaf tissue, which is a diploid tissue. And more than anything, in our strategy of doing a hybrid strategy for sequencing using ONT as the baseline and polishing using Illumina. And this is, I'm going to be now talking about the craft of my own lab in the Macombi lab in optimizing for high molecular weight DNA extractions and long read DNA fragments of over 35 kb at 36x coverage, because that has been completely available to they made this available in order to make the assembly at the highest quality. And also we have been using the latest sequencer from Oxford Nanopore, the Promethean. So we provided this to, again, our incredible collaborators, the Schatz Lab, and in particular, I'm showing in here the work of Sri, in which she is showing, using the raw sequencing of the Illumina data and actually looking at the distribution of those chimeras, it's showing that Wollemia is largely inbred given this little pattern in here in this curve. And not only that, she was able to get 21x of 40 kb long-ringed fragments in order to start off our draft from the genome sequence, followed by a number of polishing steps and followed by a number of high quality checks steps. With that, she has been able to retrieve the longest contact available in gymnosperms of 9.2 megabases. And if you want to, as a comparison from the lowly pine and the hexaploid wheat, this is truly remarkable. But again, the Schatz Lab and the Carusi Lab has shown that the genome assembly is at its highest quality. So the Kraken results, which are trying to show in here that there's a very small contamination, not even present, it's the normal amount of contamination. The highest levels when we are using our expression data, they have been able to map at the 90% and 78% in the exonic regions, but more than anything, the BUSCO results, they speak for itself because Wollemia seems to be completed at 95.3% when you compare to the embryophyta data set. And more into this, and again, talking about the remarkable work of Sri from the Schatz Lab, leveraging that expression data, she was able to genome annotate and at the gene level 50,000 genes, and almost 72% of those genes, they have a functional annotation, and close to 30, they have an unknown function, but we can actually try to live with that. and moreover, the genes that we have been able to annotate, they had been used to detect orthologs. So this is the work from the Carusi, Kolokotronis, and Varala lab, which they have been able to deploy the phylogeneous pipeline, in which they were with the aim of comparing orthogroups across the gymnosperms, showing that Wollemia has conserved almost 15,000 of the orthogroups in total from the 19,000 total orthogroups that you can see in gymnosperms. And even we have been able to see that there's a 3% absent in Wollemia. And moreover, we are starting to see some small expansions when we compare the transcription factors in this heat map, and the more clear, the more abundant. That in comparison with other gymnosperms, we start to see that these transcription factors that involve in abiotic stress are really expanded in some way. But not only it's all genes, plants are known for having large transposable elements, and in this case, Wollemia transposable elements are present at a 91.5% in this genome, and specifically the LTRs, gypsies, and I'm just going to make, I'm just hand waving to the Pollingtons, which is an ancestral viruses that happen to be over in here. But more future directions into this, which I'm pretty excited that we are currently working very hard into it, is this interplay between the transposable element, the small RNAs and other RNAs with methylation and epigenetics, and how this interplay. But even more wonderful, it has been to be able to collaborate with local experts on the Wollemi pine in Australia from Deakin University and the Botanical Gardens, in which they were able to go into the wild and isolate 5 subpopulations where they were able to resequence some of these 5 reference individuals. And we are working to see how is the diversity of this population and the Phytophthora resistance. So some conclusions is that international collaborations, including local experts, it's really crucial for conservation genomics. And long-read sequencing has helped increase in the contiguity of the assembly, and this assembly, together with substantial amount of expression data, has increased the resolution in genome annotations. And this actually high-quality genome annotation is aiding the biological interpretation of the diversity of the subpopulations, gene family expansions responsible for abiotic stress in gymnosperms. Nothing of this would have been possible without the NSF Plant Genome Research Program, which we are very thankful. And these are my extraordinary collaborators from the New York Plant Genomics Consortia and our collaborators in Australia. And I'm going to remarkably say a lot of thank yous to Dick McCombie for being our fearless leader in this endeavor. Thank you very much. I would like to take any questions if possible.
So welcome back, everyone. So I can just congratulate all of the speakers on these fantastic talks and research projects. I really, in all of them, saw a huge component of actually applied conservation, which is fantastic to see. And I guess that's the direction where we have to go with biodiversity genomics. So I see that we have a lot of questions coming in right now. So I will try to ask each speaker at least one question. But as you know, the speakers will be available afterwards in the networking session and also via Slack. So if your question doesn't get answered now, please just get back to the speakers there. So let's start with Caroline. So we have a question from Ellie. So Ellie, I really liked your work, Caroline. She's asking for the mass array design. How have you selected SNPs to maximize the information you can get from any sample for like each of the different questions with respect to individual identification, relatedness, or population structure?
Great question. So what we've been doing is we use the tissue samples that we've got from the different bilby populations to generate, looking for polymorphic SNPs across the entire metapopulation. And so that's allowed us to develop an array which allows us to look at the population structure. And then we've been developing more specific arrays for certain locations and certain sites, which are then allow us to do some more site-specific type of information. So those arrays will allow us to do things such as parentage and some other more internal relatedness type information. One thing I will say with bilby scat, it's really hard to get really decent DNA off it because obviously they're a desert dwelling species and lying out in the desert is pretty problematic, but we slowly but surely are getting there with a great team of people. So hopefully we'll finish the array soon and then we'll have something for the Indigenous communities to help understand what's happening with the wild populations.
Great, thank you very much. So next we have a question for Greg, and I got lucky enough to have my question upvoted. So Greg, I was wondering if you think that there's a risk of missing certain fine-scale genomic contributions to like any sort of adaptive phenotype and focusing on only one diagnostic marker region. So I was also thinking, given that you have this really strong signal about potential long-term changes in genomic contributions that you might not actually be able to capture in past data sets. So is it something you think we have to worry about, or what's your attitude?
I actually upvoted the question also because I was hoping that you would get a chance to answer it, because I think it's a very important question. clearly, when we're focusing on one SNP, that we have a specific question to answer, and we can't really say very much else about other factors that may be, or other phenotypes that may be have been affected over this same time series. So I think ideally we would be able to sequence the genome of all those 1,300 samples. So this was a bit of partly a cost-saving mechanism, but also because it was, we knew that we had demonstrated this SNP was linked with a phenotype that was important for conservation and management, where there had seemed to be changes. So one of our main questions was, is this also happening at the genetic level? So I think definitely we're missing a lot of things that may have been happening over that same time period for other factors, thermal tolerance or disease resistance or anything like that. But our question was focused to answer, or we focused the panel to answer this one specific question.
Yeah, thanks, Greg. That makes a lot of sense. And we can only go one step by step, I guess. The next question is to Enrique. So Camila was wondering about the really high variance that you saw in the genetic variation among your wild species, wild individuals. So she was wondering what you would associate the two, and if it might have to do something with sample quality.
Thank you. Great question. It might have something related to the sample quality. We had some variance in the sample quality for those samples, but they come from the late 70s and early 80s. And we imagine that some of that variation is related to the genetic scenery back then. So when we had higher diversity in some populations, but others the bottleneck that the species suffered might have already started. So we want to include older samples from the 60s, 50s to see if that's the case, or it's just a matter of sample quality. But yeah, we are still exploring some possibilities in that regard.
Great, thank you. Then we have a question for Ellie from Andreas Penning. So he's asking if you think that the population structure of lions across Africa will mirror the human population structure, or if the lion population structure is driven by very different geographic forces in comparison to the human population structure. Very interesting question.
Yeah, that's a great question. I think so. Just to kind of broadly speak on what we know about lion structure from the data we've had so far, there's a cluster in southern, eastern, and western Africa that splits the continent, and then there's an Asian kind of population of the Gir lions that splits the Indian lions off. I think in general, with the whole genome data, which is why we're so interested in it, there's probably a lot more fine scale structure within those three major clades that we don't really know about yet. But I do think that there could be some mirroring, although we know that a lot of the fences, so the domestic fences that keep hand, foot, and mouth disease with livestock populations that have been put up and taken down in Africa have certainly shaped kind of the way lions have been moved around, as well as the management strategies. So whether lions have been translocated, whether there's been any sort of kind of block of some of the migration patterns between countries, if there's a lot of poaching in some areas in Western Africa. So I don't think it will look exactly like it, but we could see some similarities. But I think the whole genome data will give us a lot more insight into those very fine scale structure that we haven't been able to see yet from the microsat and kind of limited SNP data that we have so far.
Great, so definitely something to follow up on, Andreas and everyone. So next we have a question for Olivia. I again got lucky with my question. I was wondering about this crazy number of transposable elements that you found in Vollemia. And I know that plants have relatively more transposable elements than other species, but I was wondering if more than 90% is normal. And if not, if you think that this could have an impact on the adaptive potential of the species. You are not unmuted yet, Olivia?
Yeah,
No.
now I'm in. I'm back. So yes, that's a great question. So in conifers and in relatively implanted species, you're expecting to have 70% on awards. I think I read a smaller question. So from that genome that it's 11.6 gigabases of data, or gigabases in general terms. You have 91, and actually a more refined aspect of this, because I'm double checking that, you know, some of those transposable elements are not open reading frames and etc. It's getting closer to 98% of the whole genome. So this is normal in plants, and it's a little bit like an outlier in that sense. Is normal in conifers. So this has been observing the Norwegian spruce and ginkgo and all of these other gymnosperms. Yes, there are potentially the drivers of the adaptability, right? And specifically, and this is why we are looking into the methylation, because I think epigenetics has to do with that mechanism of adaptation, small RNAs, and basically they're going to be the drivers of the expression, and ultimately they're going to give the evolutionary pressures. And that's basically it in regards with transposable elements. This in questions of like the variability of transposable elements, we have a slightly to start to see the SNP data from this 6 subpopulations, including the one that we have here from the New York Botanical Gardens. What we have observed is that we have one SNP every 10 kb. So that's a very, very little amount. So this is why, for instance, in one of the plots I have been showing, where you plot the raw distribution of those k-meres from the Illumina data, they just show not a lot of heterozygosity, right? So for us, it's a complete puzzle to figure out how these species are adapting and how they can retain this incredible ability to survive climate change and being there since the Cretaceous and haven't changed a bit. So for us, it's just an incredible puzzle that we have to figure it out.
Yeah, fantastic. And I guess a great model for potential alternative genomic adaptive approaches. That's great. Thanks, Olivia. So I now just went through the remaining questions, and I think one that we definitely have to ask is to Ellie, because a lot of people are wondering about how you manage such a large scale project, like organizing it, setting up the database, working across the boundaries of different countries, and so on. So maybe if you could just comment a bit on that, Ellie.
Yeah, absolutely. It's definitely been, first of all, I'll say that it's been a learning process, and Simon Morgan, who's kind of my co-lead on the project, has been instrumental in dealing with all of the permits and reaching out to the appropriate people. And I know that there's some questions about samples, how we dealt with all of those permissions, and a lot of the samples that we actually have included were samples that were already collected. We didn't want to put kind of an onus on the people that were participating to actually go out and have to collect new samples for us. That presented kind of its own challenges, because we would kind of find an existing collection, and then we wanted to actually go back to the original kind of in-country people that were involved with acquiring those samples and learning more about them. In terms of the databases, a lot of it has just been us managing it, but we've kind of found a few kind of tools that we've really liked in terms of how to process the data and things like that. For our project on tigers, we ended up using GenCove, which is based out of New York, for kind of some of the data management there, and I think that's been really helpful. But what is exciting is that through all of our kind of stumbles and figuring everything out, Simon is actually participating now in the African Biogenome Project and is on several committees. So they're he's passing on all of our kind of grassroots legwork from what we've learned trying to do this. And we're really, really hopeful that unlike with our project, which is small and species focused, that this kind of much bigger entity will be able to affect a greater amount of change within Africa than we have been able to. We didn't have kind of the ability to, pressure some of these companies to, send another sequencing machine out there so that our collaborators had immediate access to the data. But we hope that with kind of more people involved and a more kind of concerted effort from everybody, that this will be possible. So to answer your question, it was a lot of just us kind of figuring things out as we go, but trying to be as absolutely inclusive as possible, and a lot of updates and communicating with our collaborators. And I should give a shout out to Laura Bertola, who has been hugely kind of instrumental in getting this off the ground as well, who did a lot of the kind of initial legwork on Lion Genetics. So lots of people and lots to be thankful for from our collaborators. Yeah.
Great, thank you very much, Ellie. I think we have time for one more quick question to Carolyn, because people were also quite interested in your mass array approach. So have you experienced any problems with dropouts when applying the mass array to fecal samples? So that you could...
Yes.
Yeah.
lots and lots of dropout. So I should actually, I didn't say the mass array is still in development. We know it does work because we use it with ghost bats and a bunch of other species, but it's just finding the combination for the extraction that will really crack it consistently. So we're almost there with bilbies, but with any kind of scat analysis, we get dropouts. And so it's really trying to find the combination of enough polymorphic SNPs that are consistent across populations. So it does take quite a bit of work to get there, but we know once we crack it that we'll be able to use it. The other thing I should say about the mass arrays as well is they only work for a certain period of time, because obviously the polymorphic SNPs just naturally get genetic drift and get dropout happening. So they work well when you've got species with a longer generation time. I'm kind of a bit nervous about what's going to happen with a species that has breeds every 6 months. So our generation time is exceedingly short. So we'll just have to kind of watch that space and keep working with the management team to make sure that we give them a tool that's pretty robust.
Yeah, which very much ties back to what we just discussed with Craig as well, right? And maybe if sequencing costs really continue dropping, at some point we don't necessarily need these targeted approaches. But yeah, that's really fantastic work. So I fear that in 3 minutes our social networking already starts. So I will close the question and answering session with this. So now after you leave the session, please go to the networking tab, and there you will find the break time networking spatial room option. So if you choose this one, you will find most of our speakers from this conservation genomics focus session and from the previous one there, and you can discuss other questions in more detail. Or if you don't find your speakers there, just try to contact them either via Slack or via email. But with that, I would really like to thank our speakers again. I think that was a really fantastic and informative session, and I look forward to discussing more now. Thank you very much.